68K, but those are domains. The single polypeptide is 75K. And then we started to introduce the sort of experiments you do to analyze what part of this protein is on the outside and what part's on the inside. We know that the protein is 75K. We have no idea how many transmembrane domains it has, and what parts are on the outside and what parts are on the inside. So to do that kind of analysis, we use a protein called trypsin. It's an enzyme, a protease that cleaves peptide bonds. And we can think of trypsin as a common cutter that will basically cut proteins into small bits that you cannot resolve on an Estes gel. So all these peptides will run at the difront. They don't make bands. And we ask the question, okay, you have this cell right here. You expose it to trypsin, and what trypsin does, again, is it will destroy anything it encounters. It's a protein, so it cannot cross the membrane. And we've done the detergent extraction, the gentle detergent. We know that that would be a single band of 75K. And now we're going to ask the question, okay, we expose this cell to trypsin right here, wash away the trypsin, analyze the proteins, extract the proteins and analyze them on a gel. What would be in this lane right here? So what I want you to do on your handout, or you don't have a handout, but on the desk or somewhere, the handouts are coming, is put what the banding pattern you would expect right here in this lane of a cell that's been exposed to trypsin. The trypsin's been washed away, and then the remaining protein is run on this gel. And so I want you to draw that banding pattern on that gel or on a piece of paper. If you have last class handout, the gel is still on that handout. And then once you've done that, there's an Instapoll question that asks how many bands you drew and how many bands you would expect to see there. Because there's protease, it cannot, it's a protein, so it can't cross cells. It comes after arginine and lysines, after basic amino acids, positively charged amino acids. And those are so commonly found in generic proteins that anything that's exposed to trypsin will be cut into small peptides that are not resolvable on a gel. So gels cannot resolve. It'll generally cut to about, if it's randomly dispersed, it'll be every 10 to 20 amino acids. And you cannot resolve 10 to 20 amino acids on a gel. So there will not, those peptides will not be bands. They'll run at the difront, they'll run off the gel. They'll be running as if they were sodium chloride or something else that's with that information, I think, again, for another minute, discuss it, and we will vote again. And we saw a dramatic shift to the correct answer. So with someone who, so the correct answer, so you had a choice of 1 band, 2 bands, 3 bands, and 4 bands. No one in the first, I don't think anybody chose 3 bands or 4 bands. The answers settled on 2 bands or 1 band. Originally, people thought it was 2 bands, but then it shifted to 1 band. So with someone who switched their answer or was maybe convinced someone else to the correct answer of 1 band, explain their reasoning. Go ahead.
So I thought it was 2 originally, just because I was thinking that trypsin would cut off like the extracellular protein, which would leave like the 2 separate parts, so there would be 2 bands. But then I was talking to Zach, and he like convinced me and told me that since trypsin like cuts at all the peptide bonds, like that all the amino acids of that extracellular protein would be broken up and be so small that like it wouldn't appear on the gel.
Perfect. And it's not every peptide bond, but it's common peptides. So arginines and lysines are just super common. There are a few, it's sort of like restriction enzymes, if it cut between A's and T's or something like that. If y'all are more familiar with restriction enzymes, maybe you're not anymore. You don't teach that so much in molecular biology or genetics. But so I'm going to abandon that analogy. But arginines and amino acids tend to be evenly dispersed. So there are 20 amino acids, 2 of them are arginine and lysine. So you're going to cut between 10 and 20 amino acids. Those are tiny. They're going to be running off the gel right here. This is 5,000 Daltons. So it's that's going to be something like 50 amino acids or more. Okay, so what we see then is that trypsin. Let's see, maybe I even have maybe here. No, so there's no. So we would see that trypsin would cut this into tiny smithereens. Those would run off, and the remaining gel. What molecular weight did you get?
Like 60-ish.
Yeah, 60-ish, so it would run right here with the extracellular domain. So combination between this lane and this lane would tell you that there was a large 58K domain on the outside of the cell. And the reason this is protected is because trypsin cannot cross the membrane itself. To look at what's on the inside, we could do another trick, which is permeabilizing the cells. And what permeabilization means is that we have made the cells permeable. There are tricks you can do to poke little holes in the cell membrane. Sonication is one of them. A super low percentage of a very, very gentle detergent. Saponin is one of them that might remove some cholesterol. So little holes are put in there. And what that does is it allows trypsin to enter the cell. Doesn't extract the protein as we saw with a higher concentration of other detergents. Rather, it allows trypsin to enter the cytosol, and of course trypsin then will cut things up into tiny smithereens. So if we look at the gel again, and then we do a third treatment right here, where we permeabilize the cell, expose it to trypsin, get rid of the trypsin, and then I should change this to SDS gel, right, or extract it right here with a gentle detergent and run that on a gel. What is the banding pattern you would see in this lane right here? So why don't you go ahead and draw that on your gel. 2, 1. So let's just shout it out. How many bands did you find? 1. And what was the molecular weight? 2. Right. So what you're all realizing is that you destroy this guy and this guy right here. And so we have one band right here that's about 2. It's on about right here, something about there. So doing a combination of these kind of experiments is how you could try to decide what's on the outside, what's on the inside, and maybe even get a hint of how many transmembrane domains there are. You'd probably have to do more experiments because all the transmembrane domains would run at 2K. They'd be right here. Questions?
So what's the threshold for when you don't see it anymore? Because I was under the impression that if it's less than 5 kilodaltons, you wouldn't be able to see it.
Yeah, it depends on the gel, and actually, honestly, 2 you might not see either.
Okay.
So you want to look at the molecular weights right here, and then so 2 is below the lowest one, so you might not see it. But we know we can resolve 5 and 20 and 40 and 60, etc. Yeah, and there are certain different percentages of gels you can run, so it just totally depends on the gel. You can also run really long gels, so it all depends. But we only have confidence for things that run between the molecular mass markers, so I wouldn't have confidence with certainty of anything that was in this particular gel that was greater than 100 or that was less than 5, just like any other standard curve. Other questions?
I'm just trying to clarify it for myself.
Yeah.
So in each of the lanes here, for the detergent and the trypsin and detergent, they would both be it'd be one band both at 60?
For the detergent and for the, okay, so for the first one, that's what we did in the first experiment.
Yeah.
And in that one, what we did was we extracted the entire protein and ran it on a gel. So that would be a single band, and it would be 75K. Yeah. Yeah.
So when we say extract the integral protein, what does that really mean? Because if we still need a permeabilizable detergent to allow it to go through, when we say we extract it,
okay, so what we mean by extract is that is the treatment with a here in some slides I've added this, a gentle detergent. like Triton X100. So what that does is, remember we talked about micelles in the last class? That will actually extract, will take away from the membrane. It will, so the integral membrane proteins are embedded in the membrane, in the bilayer. And the micelles of detergent will actually protect the integral membrane proteins, so they'll be floating around. They'll be soluble, as extracted and floating in the buffer, protected there. Does that make sense? So look at the slides from the last class to see what that, to visualize what that looks like, that protein extracted and soluble in buffer, integral membrane protein. It no longer needs, it's no longer protected by the membrane's been. And then you take that extract and run a sample of that on a gel. Okay. So look at those slides. Other questions? Yeah.
If we used SDS instead of like the more gentle detergent, would we separate the protein into those three components?
Excellent question. If we were to just hit this cell or this membrane with SDS, would we separate the three domains of the protein? What do you all think? I see some no's and some yeses. The no's have it. And that's because, so you were shaking your head. You were very firmly shaking your head. You're right. So why don't you think SDS would separate those three domains, just hitting that membrane with SDS?
SDS is a negatively charged detergent.
Correct?
It does not disrupt the peptide bonds, but it can disrupt ionic bonds.
That's right. So it would denature the protein into one, it would get rid of the tertiary structure, so it would be a linear primary structure protein, but it would not break up the domains. The domains are attached by peptide bonds. So domains have nothing to do with primary structure. They refer to secondary and tertiary structure, how the protein's folding. Is that clear?
Yeah, thank you.
Uh-huh.
If you use detergent first and then try to
What kind of detergent?
a
The gentle one? Okay.
If you add a gentle detergent first and then try to add trypsin, can the trypsin go through that gentle detergent
Ooh.
and break up the whole thing?
I think it would. I think it absolutely would, yeah. That's a good question. And so the gentle detergent is just protecting I love that question, I've never gotten it before. So the gentle detergent is just protecting, is just adhering to the hydrophobic transmembrane domain. And the extracellular and the intracellular domain are in the buffer. So you can think of this integral membrane protein with this hydrophobic domain right there that's protected by all the detergent right there. Trypsin comes in and it would have access to both sides. Good question. I think you totally understand how this is working. Other questions? Okay, cool. So I've given you a bunch of experiments to do with trypsin treatment, with other experiments. All of these are experimental tools we use to analyze cells to figure out what's on the outside, what's on the inside, and the domain structures of proteins. And those are used daily by cell biologists. Okay, so here's the single K band. What we're going to talk about now, we're going to talk about in the next several lectures a general topic called membrane traffic. And what membrane traffic refers to is, we think of membranes as being really static in cells. We have the plasma membrane, we have the nuclear membrane, we have the ER membrane, but actually there are vesicles bringing proteins between all of these compartments. And we can think of one way of going out membrane traffic can generally be divided into the secretory pathway and the endocytic pathway. The secretory pathway is the pathway outside of the cell, going from the ER to the Golgi to the outside of the cell. That's where secretory comes from. But secreting. But secretory is sort of a misnomer because it actually, a protein doesn't have to be secreted. It can be anywhere along the pathway of the ER to the Golgi to the plasma membrane. And that's the arm that we're going to focus on today. And then later, we'll reverse direction and